Welcome and thank you for joining us today. We have a great presentation by Dr. Sarah Best at the Walter and Eliza Hall Institute for Medical Research in Melbourne, Australia. The team will share their experiences with CosMx and GeoMx research in studying heterogeneity, variance, and high-grade gliomas. If you're joining us during the 3 p.m. Pacific time airing, stay on for the live Q&A. Any questions can be posted in the comments section of the portal, and so we will be joined by Dr. Saskia Freytag, and she will help us go through your questions afterwards. But to kick us off, we're being joined by Dr. Vic Devgan of NanoString. He will share with you a bit more about both CosMx and GeoMx. Kick it off, Vic. Thank you so much.
Hello, everyone, and welcome to NanoString webinar. My name is Vic Devgan. I'm Senior Director of Marketing and heading Spatial Biology Business Unit at NanoString. Next slide. NanoString is a well-established company with a long history of innovation. We launched our first platform called Encounter back in 2008. Encounter made gene expression analysis easy and fast, especially for challenging samples like FFP tissues. We continue with innovation, and in 2019, launched our first spatial biology analysis platform called GeoMx Digital Spatial Profiler. GeoMx is the first complete automated spatial biology platform in the market. Last year, we took innovation to a notch higher and launched 2 platforms, CosMx Spatial Multi-omics and Atomic Spatial Informatics Platform. Next slide. Currently, NanoString has most comprehensive and scalable set of spatial biology solutions. We have 3 spatial platforms. CosMx SMI is the highest plex single-cell multi-omic imager with an inbuilt high-resolution microscope for readout. So you don't need any additional instruments. One instrument takes you all the way from sample to results. Whereas GeoMx DSP is the fastest whole transcriptome profiler with a sequencing readout. Although fastest whole transcriptome analysis is the most valuable attribute of GeoMx, its most differentiating value is enabling researchers to select biology-driven substructure or compartment for spatial analysis. Then we have Atomic SIP, a complete, open, and easy-to-use cloud-based informatics solution, which provides everything you need for spatial biology informatics from study design to publication. And all these 3 platforms are great on their own, but real magic is how they interact with each other, how they complement each other. Next slide. And we developed CosMx and GeoMx in-house in such a way that they complement each other for all scientific questions. CosMx is a method of choice when researchers are comparing single cells. CosMx enables imaging and quantification of high-plex RNA and high-plex protein with a single cell resolution. At the end, you will get not only single cell expression analysis and cell type cluster as UMAP, which is usually an endpoint for single cell RNA-seq experiments, but also physical location of each cell type in an intact tissue, so that you can understand how these single cells are interacting with each other or influencing their neighboring cells in their natural habitat. CosMx delivers quantification of more than 1,000 RNAs and 60 different proteins with a relatively moderate throughput of 2 to 7 slides per week. In contrast, GeoMx DSP is the best method for large cohort studies and comparing functional tissue area among multiple samples. GeoMx provides morphology marker-based tissue image, which can be used to select specific area for spatial whole transcriptome analysis. As shown here as an example, one can segregate immune compartment from a cancer compartment and get a biologically relevant differential gene expression. Since GeoMx DSP uses NGS for readout, it provides much higher throughput and process up to 40 slides per week. Next slide. Based on these complementary benefits and complementary values, CosMx is best suited for cell atlasing, cell-cell interaction, target enrichment studies, whereas GeoMx shines for biomarker discovery, tissue microenvironment analysis. And then there are some applications like therapeutic response, tissue pathology, where both platforms can be used. And when it comes to, okay, which platform is the better choice for your study, that completely depends upon your experimental requirement. Like, do you need single cell spatial resolution, or do you want quick analysis of large number of samples? And based upon that, if you need a single cell resolution, you can go with CosMx. However, if you need a quick analysis of large number of samples, or you're interested in large cohort studies, GeoMx might be the method of choice. Now, let's talk a little bit more about each of these platforms, starting with CosMx. Next slide. The CosMx is most widely adopted single-cell spatial imager available in the market, with more than 200 orders. Although we started shipping CosMx only 6 months ago, it's a battle-tested platform and has been delivered more than 150 customer projects through our technology assess program. CosMx is getting not only huge commercial success, but also good scientific recognition. As of today, we have more than 10 peer-reviewed publications from CosMx. Now, let me tell you why CosMx is winning hearts of scientific community. Next slide. First of all, CosMx gives you ability to detect more genes than any other in situ system with a large panel of 1,000 genes. It works with any sample size, whether it's FFP, fresh frozen, or tissue microarray. Has true multi-omic capability, provide high-plex RNA as well as high-plex protein analysis, with a tunable throughput of 2 to 7 slides per week. It's very important to note that when you are doing in situ analysis, plex does matter, as it is directly proportional to biology coverage, and CosMx provide the highest plex available in the market. Next slide. In addition, CosMx has broad assay menu, including off-the-shelf, ready-to-use multiple RNA and protein panels. And all the protein panels are fully validated and ready to use. And in addition to the catalog panels, CosMx also have RNA and protein customization capability. You can develop RNA panel all the way from scratch and can have a custom panel up to 350 genes. Next slide. We are expanding CosMx assay menu even further. We will be releasing 2 new panels in 2023 and add on a custom capability for protein assays. I'm also thrilled to announce that we are on track to launch 6,000 plex CosMx human discovery panel in the first half of 2024. Our 6,000 plex panel covers virtually the entire reactome. Nearly all of differentially regulated genes which matter are covered by our 6,000 plex panel. We will be starting technology assess program for 6,000 plex panel in Q4 of this year. Next slide. Now, let's change gear to our other spatial platform called GeoMx DSP. What makes GeoMx as unique and great is its ability to allow researchers to focus on real biology of tissue and get to their answers quickly. GeoMx enables whole transcriptome analysis of human and mouse species with very high sensitivity, as well as with any samples, including tissue microarray, and with very high throughput. Next slide. A unique and powerful aspect of GeoMx DSP is UV photocleavable barcodes and adaptive optics. Each RNA or protein profiling target is labeled with a unique oligonucleotide barcode attached to a target with a photocleavable linker. The barcodes can be specifically released when exposed to UV light and then collected for counting. GeoMx DSP instrument has an adaptive optics, which are comprised of 2 million micrometers, which are configurable, or you can auto-configure to any region or shape based upon your tissue biology. The ability of these micrometers to very specifically aim that UV light, so we can release only those oligonucleotides from tissue in a very specific region of the tissue. That gives a lot of power to GeoMx DSP. So as shown here, as an example here, we are showing you difference between a tumor mask and a tumor microenvironment mask. So here we show that we can first release all the oligonucleotides associated with tumor cells, shown here in yellow, and then flip that mask and release all the oligos associated with tumor microenvironment, so that the non-tumor cells. Next slide. The adaptive optics allow incredible flexibility in asking such questions with GeoMx DSP. Within the whole workflow, you first scan the entire slide or entire tissue slide, and then define region for whole transcriptome profiling or high-plex protein, which are guided by morphology of the tissue. So that you have ability to, and on top of that, you also have ability to add compartmentalization within these regions based on your biological question. Next slide. So let's take an example here. Let's say you are interested in understanding differential gene expression of the alpha and beta cells within the islets of Langerhans in pancreas tissue. With GeoMx DSP, you can get full tissue image using protein-based morphology marker staining. Next slide. Then you can visualize where islet of Langerhans are located. Select those regions for whole transcriptome analysis. Next slide. And on top of that, these islet of Langerhans can be masked to compartmentalize the alpha cells separately from the beta cells. Next slide. As a result, you will get highly specific and sensitive spatial whole transcriptome analysis based upon the tissue biology. Next slide. Now, array-based spatial technologies can be used for large area coverage, but they may miss things in the gaps or provide incomplete coverage of key pathological targets, or waste sequencing money on tissue area which are not biologically important. Next slide. In contrast, GeoMx DSP uses biology-driven ROI selection with fully automated workflow, which is powered by Leica bond here, and provide unparalleled throughput. You can go up to 40 slides per week, and all these things result in very biologically relevant, highly sensitive, and cost-effective data with a very high throughput. So, in fact, GeoMx DSP provides at least 4 times more whole transcriptome analysis as compared to any other spatial technology available in the market. Next slide. And GeoMx has a very broad assay menu. you can analyze more than 100 protein targets simultaneously on a same slide. We have more than 300 protein targets completely validated. All of our protein assays for GeoMx are completely validated and ready to use. In addition to that, we have whole transcriptome analysis for human and mouse species, as well as recently we have released canine cancer atlas panel. And there, we also provide flexibility to our customers to add on the top of this whole transcriptome any additional gene they would like to add on. And in addition to, we recently released the TCR profiling add-ons, which can be added on the top of a whole transcriptome atlas assay. Next slide. And nothing says better than peer-reviewed publication about the robustness and reliability of technology. As of today, GeoMx DSP has more than 250 peer-reviewed publications, and majority of them with the real-world FFT samples. And that shows the power and capability of GeoMx DSP. With that, let me pass this presentation to Sarah, and she will walk us through how she has been using GeoMx and CosMx in her study. Thank you, everyone.
Thank you very much for joining the webinar today. I'm Sarah Best, and I'm a laboratory head at the WeHi in Australia, co-leading the brain cancer lab with Dr. Saskia Freytag, a bioinformatician, and Dr. Jim Whittle, a neuro-oncologist. And we've brought our expertise together, and today I'll show you a bit about how we've been using spatial transcriptomics to uncover the subtype-specific heterogeneity in brain cancer. Brain cancer is a deadly disease that hasn't benefited over the past decades from groundbreaking findings that have led to personalized treatments. Glioblastoma, or GBM, grade 4 glioma, has the poorest survival and is the most frequent subtype of brain cancer diagnosed in Australia. Glioma can be stratified into two main subtypes, astrocytoma and oligodendroglioma, based on their cell of origin. These tumours progress to high-grade lesions, and IDH is a frequently mutated gene in glioma, and its mutation stratifies the lower-grade lesions, with all glioblastomas IDH1 wild type. Glioblastomas also carry a characteristic copy number change, gain in chromosome 7 and loss in chromosome 10. Recently, single-cell sequencing has stratified the fetal brain into 5 key cell states, which are conserved in glioblastomas. The composition of these cell states has recently been used to identify the heterogeneity of glioblastomas. And in this example of 3 patient tumours, we can see a great degree of intertumoral heterogeneity, finding large differences between the tumours themselves. And we can also identify the degree of intratumoral heterogeneity, looking at the proportions of each cell state within the one tumour. And we're interested in the spatial distribution and organisation of these cell states. So in our study, to investigate the spatial heterogeneity of these key tumour cell states across high-grade gliomas, we selected 3 glioblastomas and 3 IDH1 mutant gliomas. These tumours have strong expression of the tumour marker GFAP and a similar proliferative index investigated using Ki-67. We first used the NanoString GeoMx platform to investigate our samples. And to do this, we were able to use our historical FFPE blocks and annotate the key tumour regions with the help of a pathologist. We co-stained with GFAP to mark the tumour, Ki-67 to identify the cycling cells, and CD45 to identify immune cells. And then we selected regions based on our pathology annotation and segmented the cells from each region into non-cycling tumour cells, cycling tumour cells, and immune cells. And then these segments were analysed by whole transcriptomics. So these are the 6 samples that we analysed, which each contained about 5 to 7 regions of interest per sample. Pathology review from our PhD student and pathologist Oluwaseun Fatunla classified each of our ROIs, with the vast majority of these being in cellular tumour regions. And transcriptionally, we were able to confirm the activity of these different tumour regions. So regions that were involved in necrosis were identified as being transcriptionally in S phase, and this was consistent with our annotation, whereas our cellular tumour and leading edge and infiltrating tumour were all highly active and cycling. So next, we investigated the expression of key genes in IDH wild type and mutant glioma and found a strong correlation with previously published genes of interest, validating the quality of the transcriptomics from the samples. Taking the transcripts from tumour samples, Joel Moffat, the PhD student in our lab, deconvoluted the expression data into the key glioblastoma cell states I introduced earlier. Immediately, it is clear that the IDH mutant samples were quite similar and all contained significantly enriched oligodendrocytic populations, while the IDH1 glioblastoma sample displayed significant heterogeneity between the samples and contained significantly enriched progenitor and mesenchymal subsets. We next mapped the deconvoluted cell states onto different regions of interest in the tumours. And within each tumour, the IDH1 mutant were quite similar. However, the IDH wild-type glioblastoma samples had significant intratumoral heterogeneity within spatially distinct regions, such as this region in GBM3 that is spatially very close together, ROIs 4 and 5. However, region 5 contains the astrocytic population, which is completely absent in region 4. So we're really able to identify significant differences between these spatially distinct regions. Taking a closer look at the spatial heterogeneity of these localized regions, we investigated this region 4 to 6 in GBM1, where we have a gradient of tumour through to normal brain. So we modified the CNV-KIT tool to estimate copy number alterations from the transcriptome. This allowed us to confirm that region 6 is indeed normal, lacking these key alterations we identified in the other tumour regions. To use this tool to investigate intratumoral heterogeneity, we next looked at regions 1 to 3 in GBM1. And we found that tumour region 1 had a unique pattern of variance compared to regions 2 and 3, which were very similar to each other. And notably, the characteristic chromosome 7 gain and 10 loss were present in regions 2 to 3, but not present in region 1. So we think that there's significant clonal differences in these regions. Transcriptionally from this region, we saw that these 3 ROIs had significantly altered tumour proportions from the deconvolution, and that there was a significant decrease in astrocytic cell states as we moved to region 3, and an increase in oligodendrocytic cell states. And this was accompanied by differential gene expression. And interestingly, we found when we looked across other data sets that genes such as VEGF-A, which I've shown here, also carries a distinct amount of intratumoral heterogeneity across a number of other GBMs. So next, we wanted to investigate the differences between the tumour cells that were cycling and non-cycling within the same region. And so we could use the Ki67 segmentation to perform this. And so within the same region, we found significant differences between the proliferating Ki67 positive and non-proliferating tumour region. So we see that when we look at the cell states present, that only the mesenchymal in red population is similar between the 2 regions, and we have a distinct change in our astrocytic region and a large increase in the progenitor population in these cycling cells. And that a lot of these key glioblastoma mutated genes, such as EGFR, NF1, and PGFRA, were also significantly different in the cycling and the non-cycling tumour cells. When we looked more globally across all of our samples in the cycling and non-cycling compartments, we found a significant enrichment in the progenitor population in our Ki67 positive segments compared to our non-proliferating regions, where we saw a significant increase in the astrocytic population. And this really suggests that we're identifying increased tumour cell plasticity in highly proliferative regions of GBM. So next, we looked to our immune compartment, and we could see by immunostaining of CD45 that we had varied amounts of immune infiltration across our tumours. When we deconvoluted the different tumour cell states in these segments, we could identify that GBM 1 and 2 had a really high myeloid proportion and a lot of microglia and macrophages, whereas GBM 3 had a lot more lymphoid cells and neutrophils present in the tumour. And when we went back and stained for the myeloid marker CD68, we could also identify a higher proportion of these CD68 positive cells in GBM 1 and 2. And this region that's highly infiltrated by immune cells in GBM 3 is actually lacking these macrophages and microglia, which was consistent with the deconvolution data. To investigate what might be driving the different immune infiltration in these tumours, we could use some ligand receptors that were present in the panel. And we were able to identify that the astrocytic rich tumours were really driving the macrophage and microglia inflammation. And this may be due to a MIF-CXCR4 and HMGB1 have CAR2 relationship. And this is opposed to tumours that were enriched in the mesenchymal cell state, which really saw high levels of neutrophil inflammation and lymphocytes. And there was a high amount of TGF-beta signalling in these mesenchymal tumours. So to better identify which cell states and were highly correlated with each other, both the tumour cell states and the immune cell states, and also within their placement in the different regions, we performed this correlation analysis, where we can see that we've identified 3 major regions of the tumour: the central tumour, which has a high number of microglia and astrocytic tumour cells; the outer tumour, which has a high proportion of lymphocytes; and then the mesenchymal enriched tumour, where we see a high proportion of our mesenchymal cells and our monocytes and other immune cells. So to better identify this and to validate it across other gene sets, we took this Visium data that has recently been published and then was able to characterize this in terms of the different regions, where we can see our tumour region, our outer region, and a mesenchymal enriched patch. And these different regions correlated quite well with the different correlations that we'd performed. For example, in this tumour region, we see a really high density of these red astrocytic-like cells, which is what we predicted from our GeoMx data. And next, we could overlay a monocyte score onto this region and see that we have a very high proportion of monocytes present in the mesenchymal patch, which is exactly as we'd also predicted as well. So using this information from the GeoMx data from these 6 samples, we were able to postulate some different relationships that our tumour cells had with different immune cell types. And where we saw a very high correlation between astrocytic tumour cells really closely associating with microglia within the tumour, whereas the mesenchymal tumour cell states were really associated with immune cells in this outer tumour domain. And so we wanted to validate this in another platform. And so something that I don't really need to introduce to this audience, but I think it's always nice to recapitulate, is that really validating these platforms from mini-bulk to single cell. So we used the GeoMx platform, we segmented our different key cell types, and then we were able to independently deconvolute the tumour and the immune compartments. And another platform you could use is Visium, and you take the cell spots, and from each cell spot, then you could either have immune or tumor and both. And so you're doing a little bit more work to work out which cell types were. So these would be our mini bulk technologies, where we're not looking at the data from a single cell resolution, we're actually deconvoluting and kind of estimating what cell types are in what position. And then this is opposed to single cell resolution technologies such as CosMx, where we can identify each cell independently, we can find out its identity, its location, and use a 1,000 gene panel to really annotate that cell correctly. And so that's what we did. We next took our GBM sample and moved it into the CosMx platform to validate the findings from GeoMx. So this is our GBM1. This is the H&E that you'd seen from the previous example. And this isn't a serial section, it's a little bit way down the block, which is why it looks a little bit different. But we can see that from the CosMx analysis, we had 7 fields of view that we were able to analyze. And these were in similar regions to the GeoMx analysis. So we're kind of able to make some good validation between them. From our 7 fields of view, we identified 11,269 cells and just under 2 million transcripts from these. So we had a very good yield from this data. So Saskia Freitag performed the bioinformatics for the CosMx data, and this is a UMAP where we're really looking at each individual cell and where it's clustering next to cells that are most similar. And we can see that we have our major cell types identified from the data. So in red, our tumor cells, in black is our vasculature, our immune cells, and our normal oligodendrocytes. So now we're able to look more deeply into these different cell states. So looking at the tumor compartment, a further UMAP restructure of this identifies the different key cell states in a similar way to how we performed in the GeoMx. So we saw in blue this astrocytic component, in red a mesenchymal component, and in green a mixture of progenitor and neural and oligodendrocytic progenitor cells. And similar to our GeoMx data, we identified that it's in this progenitor population is really where we're seeing the most cell proliferation compared to our other cell types. So we're really carrying on this observation. Next, we dissected the immune compartment, and reclustering of this really identified our bone marrow-derived macrophages, significantly different from our microglia, our T cells, and our monocytes. And what was really nice to see was the cell identities from the CosMx and the GeoMx from the same tumor, even though they're a little bit of a distance apart. We actually predicted very similar proportions of the different immune types in these tumors. So this was very heartening, both of our deconvolution of the GeoMx and the cell annotation of our CosMx data. So plotting the different cell types back on to the H&E as we did for GeoMx, we can see that there is a significant degree of heterogeneity still across this tumor, and which we can really nicely see when we go from the UMAP of where all of these cells are and then replace them back on their cell coordinates in the tissue. And we can really see that we have some considerate clustering between these regions, where our red mesenchymal cells are really clustering together, and our astrocytic and microglial and progenitor cells are then in these other proportions. So we start to see a bit more of a domain effect in our tumor. And so we're able to investigate this domain effect and look at the tumor architecture. And so here I've just got shown the CosMx, the immunofluorescence from the CosMx, the Voronoi plots of the different annotated cell states from each of these regions. And here we have neighborhood clustering, which is a an analysis that Saskia has designed, where we can look at cells that are most frequently distributed together and call these neighborhoods and see what are in these different niches in the tumor architecture. And you can see across these different FOVs, we have different distribution of our cell types. So we can investigate who's in the neighborhood and who's not in the neighborhood. And this is using this heat map here, where we see our vasculature very strongly comes together, and we see strong neighborhoods of vasculature. Our mesenchymal tumor cells are highly clustered with our monocytes, our macrophages, and our T cells. So with these immune cells, as we saw in the GeoMx as well. Our astrocytic-like tumor cells cluster mostly with our oligodendrocyte progenitor cells, our normal oligodendrocytes, and our microglia. So there's a lot of clustering here of more tumor-intrinsic cell types that you would normally find in a brain, or that are closely related to cell types that you would normally find in a brain. We have a mixed population, and we have an oligocyte progenitor population, which has got some mixed tumor and immune cells. So next, we validated these different associations across a different data set as well. And we similarly found that the mesenchymal cells are most frequently associated with bone marrow-derived macrophages, whereas our astrocytes are most frequently found with microglia, and this is across 16 additional GBM tumors. In the CosMx analysis, we also have ligand receptors that we could investigate, and we found that the mesenchymal cells really interacted with immune cells using TGF-beta signaling, which was also consistent with the GeoMx data. So this really allows us to put together a model of the GBM tumor architecture, and really has allowed us to explore the fact that we really have a brain intrinsic tumor niche, where cells that are most similarly related to a normal brain, our astrocytic-like cells, our oligodendrocytes, and our microglia, the resident immune cells of the brain, really cluster together and are found co-localized together a lot more frequently than our brain extrinsic tumor cells, where we have our mesenchymal cells that highly interact with immune cells coming in from the vasculature, so our bone marrow-derived macrophages, monocytes, and lymphocytes. And this is in a really TGF-beta rich environment. So just to summarize what I've shown you here today, we've found that IDH wild-type glioblastomas are more heterogeneous than IDH1 mutant gliomas, but the tumor cells are very highly plastic, and especially when they're actively dividing and in regions that are actively dividing. Tumor cells arrange into distinct domains, and there seems to be more of a preference for a brain intrinsic and a brain extrinsic domain, and that immune cells cluster strongly with this brain extrinsic domain. And so this has the clinical implication that this brain extrinsic domain and mesenchymal enriched domain may be better responsive for immunotherapy or CAR T cell therapy, which would be very interesting to follow up, and whether these highly plastic tumor cells could be directed more towards a single phenotype to improve tumor cell killing in this highly heterogeneous environment, which is brain cancer. So I'll finish there, and I'd just like to acknowledge the whole team who's worked on this project. Saskia Freytag and Joel Moffat, their bioinformatic expertise has been invaluable. We co-lead the lab with Jim Whittle, and we have an excellent cohort of members of the lab and collaborators that we've worked with together. So thank you very much for your time.
Hello, can everyone hear me? We've had some audio issues earlier. Sorry about that. We do have a couple of questions, and we're just waiting for Dr. Best and Dr. Freytag to join us as well. So technology issues, so apologize for that. But thanks again for a great presentation. We do have a couple questions. If you didn't have a chance to post your questions in the chat. There should be a Q&A chat window on your dashboard for you to go ahead and put the questions in there, and we'll do our best to get as many as we can answered before we have to log off.
Hi, Lisa.
Hi, is that Kaylee or Dr. Best?
This is Sarah Best.
Oh, hi, Sarah. Great. Thanks for joining. Sorry about the technical issues here. So the couple of questions that we have, we'll jump right in. You circled back right at the end of your presentation. One of the questions that came up that I think is really great is, how did the collaboration between the clinical research team and the bioinformatics team impact this project? It sounded as though that was kind of a great partnership, and I'd love to hear a little bit more about it.
Yeah, no. So as I mentioned, I co-lead the lab with Saskia Freytag, who's a bioinformatician, and Jim Whittle, who's a neuro-oncologist. And so my background is cancer biology. So really, the three of us were really essential for bringing this project together, because we couldn't have done it without access to the patient samples and really the deep insight in the different tumor types and what to think about in terms of glioma. But I think the close connection and close discussions with Saskia, so having a research, a cell biologist and a bioinformatician kind of constantly discussing this research was really essential to ironing out and to really discovering as much as we could from the data. And it was a really fantastic relationship to work through. So I highly recommend anyone going out there to do some spatial transcriptomics to really have a really good relationship with their friendly bioinformatician.
Great, thank you. Another question that we had in here was, what was one of or many, really unexpected or surprising discoveries that emerged during your course of the research?
So I think there were two. The first one was the really discrete co-localization of the different tumor and immune cells into the different domains. And we identified this for the correlation in GeoMx and saw some hints of this from the Visium data set. But it wasn't until we really saw it in the CosMx data and saw every cell and how it was interacting physically with the other that really helped us to convince us that this was occurring. And then subsequent experiments that we've performed on more GBM samples on across more platforms, we really consistently see this pattern. And so we found that really interesting from the biology perspective. The second really interesting and exciting finding was, and I showed this, was the consistency between the GeoMx and the CosMx immune deconvolution and identification of the different proportions of immune cells. As I mentioned, these weren't serial sections. So this was, maybe 100 or 200 microns down in the tumor, even in the same region. But we really saw across the two platforms some really consistency in the proportions of immune cells that were present. So that was really exciting and surprising to see. Yeah.
Great. No, thanks for sharing that. And Saskia, feel free to chime in if you have any additional context to add. Another question, and again, you alluded to it a little bit, is the interesting choice to use both GeoMx and CosMx. Was that something that you anticipated being able to, how you were going to use both together, or was it just sort of organically developed, that partnership there?
Yeah, so it did really organically develop. This project initially started out just a GeoMx-based analysis. And we were really, really happy with that. But when we had an opportunity to validate one of the samples by CosMx, it allowed us to both really test the system, but then also to really validate at that single cell level what we were seeing from the GeoMx analysis. So it happened really organically. We've got a lot more projects running with CosMx now, but I think both of the platforms really have their serious benefits, and I think the segmentation and the whole transcriptome analysis of GeoMx is really invaluable. And then like having the opportunity to pair that then with CosMx really is what propelled the project forward. Yeah.
Great. One other question we have in here before we wrap up. What were the CD4 and CD8 T cell numbers in each tumor? And were these two cell types usually clustered together or scattered independently of each other?
Oh, wow. So I don't have the data in front of me, so I can't tell you how many, but there were few lymphocytes in general. So it wouldn't be in the hundreds, maybe let's say that. But we did see that the CD8 and the CD4 T cells were generally in a similar neighborhood. So when we did the neighborhood approach in the CosMx analysis, we often saw the T cells, both CD4 and CD8, more in the vasculature parenchymal region and not really distributed throughout the tumor body itself. So I would say from that analysis that we do see that the CD4s and the CD8s are more co-localized and really associated with the vasculature, which, you know, is how they entered the tumor. So they're not really getting much further than that, which is also really interesting in itself.
Great. Well, I think we are out of time, but thanks so much to everybody for joining us, and have a great rest of your day.